Welcome everybody. I've been asked to chair this session. Professor Hahn will be talking, and I've been asked to briefly introduce Eric, who I've known for quite some time. Eric is from the very fine vintage of 1972 at Monash University, and at that time as well he was identifiable as a very intelligent and very well suited to the medical profession. We could see this was a man who was going to be a leader and going to be a servant in his community. And I know because I was there, tucked away in the back rows, where the recalcitrant mob would stare down and watch to see what went at the front. From there he went to the Alfred Hospital, where he described his beginning training, and then to the Royal Children's Hospital, where he described being drawn into the vortex of genetics. Overseas to the UK, where he invested, I think, very wisely in some studies on Alzheimer's disease, looking to the future, is how Eric described it. Back to Melbourne, where he completed his paediatrics and clinical genetics training, and I think then South Australia made a very wise decision in 1985, because it brought Eric to South Australia, where he has remained, and I think we've been extremely lucky to have had him here as a leader within our profession and as a clinician within his field. Eric's now head of the South Australian Clinical Genetics Service, which is part of SA Pathology, and he's also a clinical affiliate with the Department of Paediatrics at the University of Adelaide. Spends most of his time these days, he describes counselling, working with families with genetic diseases. He's been president of the Human Genetics Society of Australasian International Federation of Human Genetics Societies, and in 2005 was recognised with an AO, which is a very prestigious award, which I think gives great credit to Eric and the work that he's been doing. So it gives me great pleasure to introduce a long-standing colleague to give the HDA Oration for this year, which is entitled Genetics and Disease in Childhood. Thank you very much.
Good. Thank you very much, Michael, for that very kind introduction. And Rob Norman, thank you very much for inviting me to give this year's oration. I think it's a great honour, and I'm delighted to do it. Now, strange as it may seem, we all hope to be like the man on the right, 100 years old, having outlived all our peers, reasonably healthy, retaining our faculties and perhaps a few teeth, and a sense of humour, as this man certainly has. But getting to 100, from conception through perinatal development, childhood and adulthood, is quite a challenge, and depends very much on our genetic inheritance, the environment, how the environment moulds our genes, and of course we mustn't forget chance, which is a big factor in whether we get to 100 or not. And I'd like to begin by considering our genetic inheritance. So what do we actually inherit from our parents? And I'll go through this. Mostly we inherit 2 active copies of each gene, one from our parents. We get genes that instruct cells to make proteins. We get genes that regulate the genes that instruct cells to make proteins. And we get genes that regulate the regulators, and so on ad infinitum. And indeed, I mean, if sitting next to you is a mouse, that mouse will share perhaps 97% of its genome in terms of the genes that make protein, the humble workers of the cell. But the real difference between you and the mouse lies in the way your regulatory genes are organised and function. We also inherit from our parents an X chromosome. And we get 1 active copy in males and 2 copies in females, one of which is silenced. And so you can see an X chromosome and a Y chromosome, and this ideogram shows that on the left, all the genes that are silenced on the X chromosome in women, in every cell, you make a choice, you use one X, silence the other, and those are the genes that are silenced. There are some that escape and presumably make the difference between men and women, and there are some that are actually shared with the Y chromosome. And you can actually see the phenomenon of X chromosome inactivation in the skin sometimes when a disease is present, and you can see that for example, in the pale areas here, the normal skin, the normal gene is active, and here in the pigmented skin, the gene which has the gene abnormality is active and demonstrating that one has been silenced in each cell, with each of these stripes coming from a single progenitor cell way back in early development. We also inherit a group of genes called imprinted genes that represent about 1% of our genetic makeup. And the best way to think about them, I think it's quite hard otherwise, is to think in terms of the disorders they cause when imprinting has failed to occur properly. And these two disorders, Prader-Willi syndrome and Angelman syndrome, are the two paradigmic disorders that are associated with imprinting errors on chromosome 15. And so this young man has Prader-Willi syndrome. He would have been a floppy baby towards the end of the first year of life. He will have perhaps started to eat a bit more than he should have. He'll be mildly delayed in development. And all of those features are the result of a situation where normally the genes on that part of chromosome 15 that he inherits from his mother should be switched off, and he should use only his maternal genes. And the disorder occurs when father's copies of the genes are not expressed. And Angelman syndrome is the converse. It's a similar region on chromosome 15, and the disorder occurs when the mother's copies of the particular genes are not expressed. There are also mitochondrial genes, a separate set. They're not in the nucleus of the cell. They're situated out in the cytoplasm. They do a whole range of functions, one of the most important being to produce energy, and clearly a very important part of our genetic makeup. In amongst all of those genes, there is a huge amount of genetic variation. And there are simple changes in the genetic code, so-called SNPs, where one little fragment of the genetic code changes from one thing to another without necessarily causing a major problem in gene function. There are repeat sequences, so-called transposable elements, and most recently people have discovered so-called copy number variations where large chunks of the genome can be missing or duplicated, and I think one major push in terms of research nowadays is to try and understand exactly what that means. And so, in summary, from our parents we inherit paired genes, sex chromosome genes, imprinted genes and mitochondrial genes. Amongst those genes are genes that are really important for normal function, and where having a fault in one gene is enough, almost on its own, to cause a disorder. And when genes like that fail, you get the common disorders of childhood that are serious, well known to everybody, like cystic fibrosis and beta thalassemia, spinal muscular atrophy, where one particular gene has failed and a serious disease occurs, and occurs typically early in life. But again, there's complexity, and it's good to think how genes and environment interact in this sort of situation, because it's never as simple as there being just one gene as the cause of a particular health issue. And so you can go, as it is on this slide, through from a situation where perhaps one gene really is almost on its own the cause of the problem, like thalassemia, to situations where one gene, perhaps with some modifier genes, cause spinal muscular atrophy, where the main gene clearly causes a disorder, but there are modifier genes which determine how severe that'll be. Then there are the common malformations, cleft lip and palate, spina bifida, congenital heart disease, where variations in several genes create the predisposition to the disorder. Then you can get more complex situations where you've got one main gene, the environment, and modifier genes like cystic fibrosis and PKU. You can traditionally argue, is PKU a genetic disease or environmental disease? If you don't have the genetic problem, you don't have the disease. But if you don't have phenylalanine in your diet, you don't have the disease. And so it's a clear interaction between diet, environment, and genetic makeup. And the same is true of cystic fibrosis. No infectious disease in your environment, the disorder is completely altered.